Hello and welcome everybody to my channel Omni Bioentrance, and today I will be discussing CSIR PYQs of December 2023 shift 1 or the morning shift. I have discussed cell biology, applied biology methodology, DNA replication, protein synthesis, genetics, biochemistry. Sorry, this is part C question discussion and this is part 2 of the discussion. If you are new to my channel, please do like, share and subscribe. So coming to the first question. Given below is the structure of a gene whose transcription is terminated in a row independent manner. When the terminator is operational, here is the terminator, the short transcript of 150 bases is formed. This is the short transcript of 150 bases. And when it is not operational, so when the terminator is mutated or it is not operational, a longer transcript of 200 bases is formed. So this is the 200 base pair base transcript. A researcher generated several mutations in the terminator region. This is the terminator region. So he has mutated the terminator region and examined the transcripts obtained. The manipulation done are. So before coming to that, we will see what is row independent termination. So in a row independent termination, the row independent terminator contains this is the inverted repeat sequence and a string of polyadenine. These are the inverted repeat sequences and a string of poly-A. This is the terminator region of the DNA. The inverted repeats are transcribed into RNA. So this is the RNA. The inverted repeats are also transcribed. This is transcribed and this is also transcribed into RNA. Along with that, the poly-A region is also transcribed and you get poly-U in the mRNA. So the string of U's causes the RNA polymerase to pause. And inverted repeats in the RNA fold into a hairpin. The diagram that I have drawn is not that proper, but anyway they will form base pairing, complementary base pairing, and you will get a stem loop structure. Sorry, a hairpin structure. So you will get a hairpin structure over here by complementary base pairing of the inverted repeats. This will destabilize the DNA RNA pairing. So poly-U and the hairpin loop, they will destabilize the RNA DNA pairing and the RNA transcript separate from the template terminating transcription. So this is what is happening in row independent termination. Now what are the mutations that the researcher has done? And what is the result that you will get? 3 nucleotides of the string 8Ts were replaced by GCC. 3 nucleotides of the string 8Ts. 8Ts means it is on your DNA strand. So the 8 out of the 8 T's, 3 T's have been removed and they were replaced by GCC. So instead of poly-A tail, you will not get a poly-A tail in the second strand of DNA, and as a result of that, after transcription, the mRNA will not get poly-U tail to terminate. So what will happen is the terminator region got mutated, so poly-U tails are also not happening. So you will get a long transcript in the case of A. The 8T sequence was transferred to the template strand. So instead of A over here in the template strand, they have placed it with T. So when T comes, the RNA transcript will be A. So it will not be poly-U again, it will be poly-A on the RNA transcript. So that will not result in termination. So you will get a long transcript in the case of B. The sequences that generate the paired stem were altered to disrupt the pairing. So the sequences that generate the paired stem. So that is this. This is the hairpin loop. That is the paired stem. They were disrupted. That is, these inverted repeat sequences were disrupted. So the hairpin loop will not be formed, and RNA synthesis will continue. So here also you will get a long transcript in C. Then D, if you see, the sequences that generate the paired stem were altered to disrupt pairing, followed by introduction of compensatory mutation to restore the pairing. So you have mutated it as well as restored it with another compensatory mutation. So naturally short RNA transcript you will get, because here it will be termination of mRNA synthesis in case of D. So long transcript in A, B and C and short in D is the answer. So the answer here will be option 4. Then next one. Following statements are made about initiation of translation in eukaryotes. The EIF2 facilitates correct recognition and binding of ribosomal subunits. EIF2 guides initiator tRNA to the ribosome. So first statement itself is not correct. You have to see for combination of all correct statements. Then EIF2B activates EIF2 by replacing its GDP with GTP. That is correct. EIF3 binds to the 60S ribosomal subunit. EIF3 binds and is associated with 40S ribosomal subunit, and it plays a role in keeping the large 60S subunit from prematurely binding. So here they have given as EIF3 binds to the 60S ribosomal subunit. No, it is 40S ribosomal subunit it is associated with. So C is not correct. EIF5 promotes association between the 60S ribosomal subunit and the 48S complex. That is correct. The EIF6 bind to the 60S ribosomal subunit and block reassociation with the 40S subunit. That is also correct. Now if you know that B is correct and C is wrong, then you can happily go for option 2 even if you don't know about D and E. So answer here is option 2, that is B, D and E. Then next one. Following statements were made about rolling circle mechanism of replication. So rolling circle mechanism of replication we will see. So nick is formed in the circular, this is the circular double-stranded DNA. Nick is formed there by an endonuclease. You get a 3' end and a 5' end. The 3' end is elongated just like the leading strand. This 3' end, it is elongated just like the leading strand, whereas the 5' strand is displaced and is synthesized as lagging strand with a series of Okazaki fragments. And this will keep on rotating. That is rolling circle method of replication. Now we will see what they have asked. They have asked to choose the combination of all correct statements. So it occurs unidirectionally, yes, with only one replication fork. That's all. That is correct. The E. coli phage CSIR 174 phage uses this mechanism to replicate double-stranded single circular genome. The CSIR 174 phage has single-stranded DNA, not double-stranded. So that is also not correct. E. coli utilizes this mechanism to replicate its double-stranded DNA genome. So E. coli uses theta replication, not rolling circle mechanism to replicate a double-stranded genome. Then In the lambda, the progeny DNA may range several genomes long before it is packaged. So here what happens is lambda also shows rolling circle replication. When the virus, I will tell you from the beginning, just a short brief about where rolling circle replication comes in lambda. So the virus, it will attach to the host cell and eject its DNA. The DNA circularizes at cos sites and it undergoes theta replication first. Then it undergoes rolling circle replication producing linear genome concatenamers. Then DNA is packaged into the capsid. Then cell lysis follows and the phage progeny are released. So D is correct. The lagging strand is not formed in rolling circle mechanism of replication. Lagging strand is also formed in rolling circle mechanism. I explained it in the diagram. So A and D are the correct answer. Answer is option 2. Then next one. A portion of the M13 encoding mRNA encoding protein is shown below with the start codon underlined. So AUG, here it is. This is the start codon. Which one of the following tRNAs is most likely used for translating the second codon in the open reading frame of the protein? So second codon is UUG. So this is the tRNA, this is the mRNA, this is UUG, and complementary base pairing you will get AAC. Sorry, CAA, 5 prime to 3 prime. So it is CAA that you will get. So if you see the diagram over here, you don't see any CAA in the 5 prime to 3 prime end. Okay, even if you consider wobbling, that is third base of the codon and the first base of the anticodon. This portion, even if you see, it can undergo wobble base pairing. Even then you will not get the correct answer in here. So as per me, no option is correct. Then before coming to the next question, I will tell you what is zipper and trigger mechanism by which bacteria enter into the host cell. So this is zipper mechanism. Zipper mechanism is used by Listeria, Escherichia and others, whereas trigger mechanism, Salmonella, Shigella are using it. So what happens in zipper mechanism? So Escherichia species, they encode invasine. This is the bacterial invasine protein. They bind to beta-1 integrins on the cell surface. These are the beta-1 integrins on the cell surface. The bacterial invasines bind over there very tightly, and this will induce actin polymerization. This is the actin filament. It is getting polymerized, and the host cell membrane will it will wrap the surface of the bacterium and engulf it. That is zipper mechanism. Now coming to the trigger mechanism. In trigger mechanism, certain soluble effector proteins, bacterial effector proteins are injected through type 3 secretion system. These effector proteins will polymerize the actin filament and you will get ruffles. These are the ruffles of the membrane that will engulf the bacterium. That is happening in case of trigger mechanism. Coming to the question. Bacteria employ various mechanisms to invade or enter host cell, which are either phagocytic or non-phagocytic in nature. Given below are some of the mechanisms which are generally used for carrying out these processes. Which one of the following represent the combination of correct mechanism for invading non-phagocytic cells? Some bacteria express a protein called invasin that is recognized by host cell beta 1 integrins. That I already told just now when we did zipper mechanism. So that's correct. Actin polymerization along with the assembly of clathrin coat results in the internalization of bacteria by zipper mechanism. That's also correct. Some bacteria including Salmonella enterica use trigger mechanism to inject a set of effector molecules in the cytosol through type 3 secretion system. That is also correct. These are the 3 points that I explained just now. Then D, if you see some bacteria attached to the host cell surface receptors inducing local elevation of Ca2 plus in the cell cytosol leading to fusion of lysosomes with bacteria containing plasma membrane vesicles. Now if bacteria fuses with lysosome, then it will naturally result in death of the bacteria and it will not be able to invade into the host cell. So D is not correct. So A, B, C is the answer. Then next one. Next one is GPCR. So in GPCR, in the inactive state, G protein coupled receptor, that is GPCR. In the inactive state, it is bound to G alpha, gamma and beta subunits. G alpha is bound to GDP. But when it binds to a ligand, the G alpha subunit get detached from the beta gamma subunit. GDP get hydrolyzed to GTP. And this G alpha subunit, it will activate adenyl cyclase. Adenyl cyclase catalyze the synthesis of CAMP. CAMP will activate pKa. Now how does pKa get activated by CAMP? So the inactive form of pKa, it has got 2 catalytic subunits and 2 regulatory subunits. The tetrameric form, it is inactive. When CAMP binds, that is CAMP, the 4 CAMP are binding here. When they bind to 2 sites on each of the R subunit, the R subunit undergo conformational change and it will dissociate from the catalytic subunit, and the catalytic subunit is free now and you will get an active pKa. Now what does the active pKa do? It will activate a molecule called CAMP responsive element binding protein or CREB, and it will phosphorylate it, which modulates gene transcription. So this is what is happening. Now we will see the question. The CAMP, pKa, CREB. CREB, I told you it is CAMP responsive element binding protein. Pathway regulates many important biological processes from hormone synthesis to inducing long-term memory in the brain. The following statement describe the effects of mutation in the components of the pathway on gene transcription by CREB. Loss of which of the following statement is incorrect? So you have to see for the incorrect statement. Loss of function mutation in a CAMP binding site of the pKa regulatory subunit leads to the inactivation of gene expression. Now if there is a mutation, that is loss of function of CAMP binding site on the regulatory site of the pKa, that is over here, CAMP will not bind there. So pKa will be inactive. pKa inactive means gene expression is not happening. It's not going to happen. So A is correct. Then B. Activating mutation in the GTP binding domain of the alpha subunit of GS leads to the activation of gene expression. Activating mutation in the GTP binding domain of the alpha subunit. That is over here. This is the GTP binding domain of the alpha subunit. It leads to the activation of gene expression. Yes, alpha subunit always bound to GTP means it will naturally result in adenyl cyclase activation, which will result in the synthesis of CAMP, and CAMP will activate pKa, and pKa will result in gene expression activation. So B is also correct. Then inactivating mutation that prevents the regulatory subunit of pKa to bind to the catalytic subunit lead to the activation of gene expression. So inactivating mutation. What does that mutation do? It prevents the regulatory subunit of pKa binding to catalytic subunit. So regulatory subunit will not bind to the catalytic subunit. The catalytic subunit will be free. pKa will always be active. Active pKa means gene expression. So C is also correct. Inactivating mutation in the pKa phosphorylation site of CREB leads to the activation of gene expression. So inactive pKa, what it does is it activates a molecule called CREB by phosphorylating it. So here inactivating mutation in the pKa phosphorylation site of CREB, that means it is not getting phosphorylated. What CREB is not getting phosphorylated. It leads to repression of gene expression, not activation. So answer here is, they have asked which of the following statement is incorrect. D is incorrect. So answer here is option 3, only D. Then next one. Extracellular matrix comprises of various proteins and polysaccharides that assemble into an organized meshwork. This associates with the cells that produce them. Given below are few statements regarding different components of the matrix. Which of the following option represent incorrect statement? Collagen is the major protein of the extracellular matrix and is long and triple-stranded helical structure. That is correct. Hyaluron, which is produced in large quantities during wound healing, is a type of glycosaminoglycan. That is correct. That contains sulfated sugar, not sulfated sugar. It should be non-sulfated sugar and is covalently linked to the core protein. So B is the incorrect statement. Now we will see C. Syndicans are plasma membrane proteoglycans that interact with the actin cytoskeleton and signaling molecules of the cell cortex. That is correct. Decorin is a small proteoglycan secreted by fibroblast and contain 1 to 10 gag chains. That is also correct. So which is the option that is not correct? Only B. Answer is option 4. Then next one. Apoptosis refers to programmed cell death that is triggered by specialized intracellular proteases called caspases. The intrinsic pathway of apoptosis. So this they have asked here only about intrinsic pathway. Nothing has been asked about extrinsic pathway. Which of the following combination represents all correct statements? So A. Depends on the activation of Fas domain by the Fas ligand, which activate DISC. A itself is wrong. That is extrinsic pathway. It happens in extrinsic pathway. So first option is not correct. Is regulated by the BCL2 family of proteins. That is correct. Consists of a key regulatory protein APAF1, which is water soluble component of the electron transport chain. APAF1 is not a component of electron transport chain, whereas cytochrome C is component of electron transport chain. So C is also not correct. Recruits pro-caspase 9. So 2 is not correct. 3 is also not correct. Option 2 is not correct. 3 is also not correct. Option 1 is also not correct. So what is left out? B and D. Okay, we will see. D. Recruits pro-caspase 9 into the apoptosome, and once activated, caspase 9 cleaves and activates downstream executioner caspases. That is correct. So answer here is option 4, that is B and D only. Then next one. The following represents a southern hybridization of restricted genomic DNA probed with a DNA fragment corresponding to gene Z. Z is a single copy gene. The hybridization patterns of the parents and their progeny have been presented. Which of the following option is correct interpretation of the observation? Gene Z is present on an autosome. But here if you see the male parent and the male progeny, they are showing 2 bands, and the female parent only single band. So it is not present on the autosome, it is somewhere related to the sex chromosomes. Or gene Z is an example of X-linked gene. We cannot say that from the data given here. Then, in this organism, the female is the heterogametic sex. That is correct because it is having only one band. So it should be Z0. And the male is homogametic sex, Z. That is why 2 progeny, 2 bands. Whereas female is having only one band corresponding to Z gene. So it should be Z0 for female and male would be Z. So that is correct. 3 is correct. Polymorphism of this gene has been studied by RAPD. RAPD doesn't use southern hybridization technique. It uses PCR. So 4th is also not correct. So the answer here is 3. Then next one. Epistasis is observed between different genes in a flower color of sweet pea. A red variety on selfing yields red is to white in the ratio 9:7. The precursors and intermediate give white color. The following biochemical pathways were proposed for the above observation. 9:7 means it is complementary gene interaction. So complementation between 2 non-allelic genes are essential for the production of a particular phenotype. Now this is white colored sweet pea flower where the capital C gene is mutated to small c. And this is another white colorless sweet pea flower where capital P gene is mutated to small p. So once you cross both of them, you will get purple color, not white color. Why? Because capital C and capital P, both genes are present and they will complement each other and you will get purple color instead of white. Now F1 on selfing, you will get 9 purple colored and 7 white colored plants. In the F2 showing complementary gene interaction. Now this is the precursor for flower color. Gene C, it will produce enzyme C, it will produce intermediate chromogen. Gene P will produce enzyme P and you will get anthocyanin or purple coloration. This is what is happening in complementary gene interaction when both C gene and P gene are present. But if one of the gene is mutated, you will not get anthocyanin color. If P gene is mutated, then also you will not get color. If C gene is mutated, then also you will not get color. If both the genes are present, you will get colored flower. Now here, instead of the purple color, it is the red colored product. Now first option A, precursor gene A, intermediate X, gene B, final product red. That you will get complementary gene. Both the genes are present. Active, not mutated. So final product red you will get. Then precursor extracellular gene A, precursor intracellular gene B, final product red. Both the genes are present, active, not mutated. So naturally end product will be red. Now precursor gene A or gene B, then it is not going to work out. You will not get red color. Either gene A or gene B means you will not get red color. You will not get the 9:7 ratio. So both A and B are correct. Answer option 2. Then next one. This is a hypothetical example. In a plant, a single gene governs flower color. The wild type color is red while the mutant is white. It was demonstrated that insertion of a transposable element caused the white phenotype. In a PCR test, a set of primers outside the site of insertion is used to amplify the genomic DNA, and the PCR products are resolved by agarose gel electrophoresis. A genetist made a cross between two plants. 30 progeny from the cross was analyzed by PCR. Each lane in the gel below represents analysis of one progeny. So in a plant, single genome governs flower color, and the wild type color is red and the white is mutant. Now how did the white become mutant? Because of the insertion of a transposable element. In a PCR test, a set of primers outside of the insertion site is used to amplify the genomic DNA, and the PCR products are resolved on agarose gel electrophoresis. So now what did the genetist do? He made a cross between two plants, red and white. 30 progeny of the cross were analyzed by this PCR test, whatever they have told here. And each lane in the gel represent analysis of one progeny, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 till 30. So based on the above, which of the following statement is correct? So before coming to that, I will tell you what is a dominant marker and a codominant marker. Codominant markers, they can clearly discriminate between the homozygote and the heterozygote. See here it is parent 1, parent 2. This is F1 progeny. So capital A, small a. You will get a progeny as capital A, small a. Both the bands will be seen. One is of capital A, another is of small a. That is case of codominance. Now dominant marker means these are the parents P1, P2. F1 will capital B, small b. It will show only the dominant band, not the recessive band. So that is in the case of difference between a dominant and a codominant marker. Now in this case we will take the upper band as mutated ones and the lower band as non-mutated ones. Okay. So because why we take upper band as mutated ones? Because in that transposable element has got inserted. So that is the mutated means they are white and the lower bands are red. The DNA marker is dominant. The DNA marker is not dominant. You can see both the bands, mutated as well as non-mutated. Then you have here only the non-mutated one. Then here you have the mutated one. So all kinds of progeny are seen here. So it is not a dominant marker. Plant 3 has white flowers. Plant 3 is this. Plant 3 is not mutated. It is a dominant one, that is wild type, that is red color. So it is a lower band. So it is not mutated. So 2 is also not correct. In the above cross, one of the parent was heterozygous while the other was homozygous. If this is the condition, as you see here, capital A, small a, capital A for the red color, small a for the white color. So they have told one parent was heterozygous. So capital A, small a, small a, small a. So you get only capital A, small a, small a, small a. These are the bands you should get. But you can see the band of capital A also. So option 3 is also incorrect. If here both the parents must be heterozygous, not one is heterozygous and other is homozygous, then you will get all the 3 combinations as seen here. See here, if it is capital A, you will get capital A band, capital A band and small a band also. So both the parents have to be heterozygotes. Then, if the progeny from the cross between plant 1 and plant 30 is analyzed in a similar way, 2 different kind of banding pattern will be observed. Plant 1 is heterozygous, capital A, small a. Plant 30 is dominant one, so capital A, capital A. So you will get 2 different banding patterns, capital A, capital A, small a. So fourth statement is correct. Answer is option 4.